additional charts up that were not this morning. So there's this one's probably the easiest to understand of the models. In a sagittal section, we have this upright model here. And then we have a smaller upright model here that just doesn't have as much detail on it. So the scrotum is, or scrotal sac, you can use either term. That's the skin of the sac that encloses the testis. That would be letter A on your diagram. Now on some of these models, there's a little dotted line just deep to the skin, representing the smooth muscle of the scrotum, which is the dartos. I talked about that in lecture, and you need to know that for lecture, but it won't be something I'll have you tag for lab. Okay? It's a little bit of a dotted line. It's also shown on this one. Is the dotted line in that gray area. That's the dartos, which is the smooth muscle. The muscle that you need to know for lab is the premaster muscle that we'll get to in just a moment. The testis itself is B. And then on the right-hand diagram is a diagram of the different tubes and pathways. That's shown in the upper left over here, but I never have this chart out on the actual lab exam because there's just too many words scattered all over the place to try to cover up. All right? It's a pain just covering the rest of these charts. So instead, you know, sometimes I have a diagram in one of these sheets, like the lingual frenulum. So there'll be, I don't have it with me now, but there'll be a couple of pages like that. One showing the seminiferous tubules and straight tubules and efferent ducts, just like you have in your chart. And the other one showing the cells of the seminiferous tubule and interstitial cells. Okay, you have a similar diagram. So those two components will be out on the exam, but just not this chart. So I'll have a diagram similar to the one in the upper right. And D is representing the tunica albuginea, or the capsule. Divisions from that capsule divide the testes into lobules. So the septi are E, and the lobules are O. There's two O's, so call that O1, if you will. And then P would be the seminiferous tubule. And that would be Q, sorry. We have three P's. No, F, I read it wrong. F is the seminiferous tubule. Closed loop. It's the seminiferous tubule. Down the lower right is a diagram of the seminiferous tubule. And so O2 would be the sustentacular or Sertoli cell. It's actually the nucleus of the cell, because the cell, remember, is the entire, forms the wall of the seminiferous tubule. So O2 is really just pointing to the nucleus. And then P2 would be the developing spermatocytes. That's down the lower right-hand diagram. And the interstitial cells would be letter N. Those are endocrine cells making the steroid hormone testosterone. And because they make it from cholesterol, which is a lipid, they would have a lot of smooth ER. Because that's what they use, the organelles that are used to synthesize steroid hormones. All right, then G, back to the upper right-hand diagram, would be the straight tubule. All the seminiferous tubules come together and form a straight tubule. H would be the reedy testes. And I, the efferent duct or efferent ductules. And then you see the epididymis. So the epididymis is found on the posterior aspect of the testis. And we start out with the head, which is J, and the body, which is K, and the tail, which is L. So remember in lecture this morning, well, you may not remember because I covered a lot, but the tail is the most significant region because that's where the sperm mature and develop the functional enzymes for the tails to beat. They don't actually beat like this. They kind of look like an egg beater. They kind of go in a corkscrew fashion. And then the enzymes covering the nucleus are capable of digesting the protein from around the egg. So that takes place in the epididymis. So if we have a guy who's had a spinal cord injury that's say around T12 or even L1, his autonomic nervous system would still allow him to have an erection, emission, ejaculation. He could get his partner pregnant if they so desired. He wouldn't have any feeling, all right? He wouldn't enjoy the process as far as an orgasm is concerned, but they could still have intercourse and the sperm would still be ejaculated. However, if the most distal portion, the narrow portion of the conus medullaris is damaged, the sacral region of the spinal cord, then his parasympathetic portion of the autonomic nervous system from that region wouldn't work and he wouldn't be able to have emission or part of ejaculation, which is it's mixed, smooth and skeletal muscle. So they would, if he and his partner wanted to get pregnant, then they would have to do an aspiration, go in with a needle and withdraw sperm, and they would do so from the tail. If they took it from higher in the epididymis or from the seminiferous tubule itself, that sperm would not be able to swim to the egg, and once it got there, it wouldn't be able to digest its way through to the membrane itself of the egg. Though there is a procedure now where they can take one sperm, a very long, thin glass pipette, and inject just one sperm into the egg. And that will trigger the egg and the sperm to unite and undergo mitosis in about 50% of the cases. They use it primarily for sperm that are lacking tails or lacking an enzyme and the tails don't beat. And so putting them in a petri dish together wouldn't help. All right, so the epididymis then at the tail then forms the vas deferens or ductus deferens. And that is letter M. And it leaves from the scrotal sac and passes into the spermatic cord, which is the bundle of the vas deferens and the blood vessels, passes through the superficial ring and deep inguinal ring, lateral to the bladder, and then to the posterior surface of the bladder. So probably the best diagram for the structures that you have there is on that chart or over here on this chart. So this spermatic cord has been opened up. Here you can see the covering. So here would be the superficial inguinal ring, and the deep ring would be on the far surface. So in your diagram, the spermatic cord is formed of, I think C is supposed to be the spermatic cord, that bar, including everything in there. The cremaster muscle is R, which doesn't help you on that diagram, but you can see a good cremaster muscle right here. Remember that skeletal muscle derived from the internal abdominal oblique. On the models, you can see it on this one probably best. Here's the superficial inguinal ring, and this brown layer right here that has the lines in it is supposed to represent the cremaster muscle. Okay. Then if we cut that open, we can actually see the components of the spermatic cord, which include the vas deferens and the testicular artery, and this blue structure that you see here, as well as over here, that reticular network. In your lab manual, it's called the testicular vein. Please cross that out and change it to pampiniform plexus. Testicular vein is not the best answer, so you would get half credit. What's it called? Pampiniform plexus. P-A-M-P-I-N-I-N-I-F-O-R-M. Pampiniform plexus. Let me take this up already on the board. So it's just, we talked about the heat exchange.
So is that just for the vein?
That's just for the vein. Okay, that would be T, and the artery is S.
I'm sorry, what was
T. Then V is showing the internal inguinal ring or orifice. You can say deep inguinal ring or deep inguinal orifice or internal or external, okay, but be sure to include the word inguinal. So whether you say orifice or ring or superficial or external, that doesn't matter, but include the word inguinal. All right, so V is showing the external or superficial, and that would be this arch right here. And on the chart, it would be this arch right here. And on that chart over there, they actually have two little rings. So on the lower portion, sorry. So right here, so right here would be the superficial or external, and there would be the deeper of actual. Go ahead and lay it down on its side because it's missing one leg. It's going to balance. All right? Yeah. So superficial or external, deeper internal. And so this is the vas deferens and tympaniform plexus, testicular artery passing through that. And then the gonadal vessels are going to continue there, whereas the vas deferens is going to come around to the bladder. This right here would be the inguinal ligament.
So it's like,
Yeah.
like this.
Yeah. So, all right. The suspensory ligament, I did mention in lecture, but it's a band of connective tissue. It's not muscle, it's just collagen fibers. This is a cross-section through the pubic bone. Here's the corpus cavernosum, and it's just suspending, it's attaching. Let me use your pelvis again. So, right here, there's a band of connective tissue that's going to attach to the penis and hold it up to the bone there so it doesn't come down like that. So that's the suspensory ligament. And that would be letter U on your diagram. All right, back to the vas deferens. Is letter M. As it passes behind the bladder, it joins with the seminal vesicle, as we'll see on the next page, and the two form the ejaculatory duct. The ejaculatory duct is P1 over here, best seen passing through the prostate gland. So on that sagittal view, or on our sagittal view on the model, is the best view. So here we have the vas deferens coming around to the back of the bladder. This would be the seminal vesicle, and the two join together to form the ejaculatory duct that passes through the posterior part of the prostate gland. All right, so we have three glands, three pairs of glands. We have the seminal vesicle. We have the prostate gland. The prostate gland is found between the bladder and the external urethral muscle, or the urogenital diaphragm. And then the third gland is found in the inferior portion of that, the vulvourethral or Cowper's gland. So here's seminal vesicle, here's prostate, and here are the two small vulvourethral glands. So those can be seen on the next page, 214, 215. So this is a posterior view of the bladder. Keep leaking urine into the pelvic cavity here. So here are the ureters. Medial to the ureters are the vas deferens, or the ductus deferens. Lateral is the seminal vesicle. And then they join together within the prostate gland. This is the prostate gland to form the ejaculatory duct. The prostate gland itself has its own openings. All right. So if we lift this out again, here would be the prostate gland. It's a donut-like structure wrapped around the prostatic urethra. So on your diagram, then, A is the vas deferens, B is the seminal vesicle, and D is the prostate gland. C is supposed to be pointing to the ejaculatory duct, but you're not seeing very much of it. And then in the diagram below, E would be the prostatic urethra. H is the urogenital diaphragm, which is not, there's no name for it on 15, but the urogenital diaphragm or the external urethral sphincter would be H. And then passing through that, the narrow portion of the urethra is called the membranous urethra. That would be G. And then K would be the penile or spongy urethra. Either term is fine, but if you call it the spongy urethra, it makes it easier to remember that it's in the corpus spongiosum rather than in the corpus cavernosum. Again, it just helps to keep it straight. So when we look at the penis itself, there is no cruce of the penis on your lab manual because all of our models are just sagittal sections, but this would be the crus that I diagrammed in lecture. And I'll actually model it for you with clay on Wednesday. But down here would be the corpus cavernosa. And so on our sagittal sections, corpus cavernosa is going to be I. And remember I said it's a dorsal structure. So again, on a cross section, remember the monkey face I talked about in lecture? So those are the two corpora cavernosa, and then ventral to that is the corpus spongiosum that contains the urethra. So it's labeled that way rather than anterior and posterior because of the erect position of the penis. So if the penis was hanging down like this, these would be the two corpora cavernosa and the corpus spongiosum. But in an erect position, then it becomes more horizontal, slightly vertical, and so these are dorsal and this is ventral. So the two corpora cavernosa are dorsal. We're only seeing one, and that would be this structure right here. And that would be letter I in your diagram.
What did you say H was?
H is the external urethral sphincter or the urogenital diaphragm. Now you'll notice that there are septi in this model of the corpora cavernosa. And these can be ruptured. So people talk about a boner, but there really is no bone in the human penis. There is in the male. But when these septi are broken, then the penis are torn. The penis can be described as broken. So you can have a broken penis even though it doesn't have a bone. Usually develops because of some different sexual behavior or people that are trying to act like circus animals. I was talking about this with my students at AR, and we looked it up online, and they gave the description of this couple. She was on a chair or a couch, and he was trying to land on her from about 10 feet away and make the insertion of his penis into the vagina, and he missed and hit her pubic bone, and that bent the penis sharply and ruptured the septi, which then bled into the corpora cavernosa, and so his penis was like tucked over, bent over on one side. That's a broken penis, okay?
So what happened? You wouldn't believe me if I told you.
Exactly right. So then below that, this would be the bulb that I described in lecture. It's not on your lab packet, but then that becomes the corpus spongiosum. There's only one of those, and it contains the spongy urethra. At the distal end of the penis, it expands like a cap and forms the glans penis. Okay? The glans penis is exposed when the prepuce is pulled back. So on this particular model, there's the prepuce or foreskin, but you can see a little bit of the glans penis. All right? And on this one, the prepuce is farther extended. So in a male circumcision, the prepuce is cut back here near the base. We'll talk about that when we talk about female circumcision on Wednesday. They're two entirely different things. But for male circumcision, it's supposed to make it easier to keep the gland area clean. Certainly, kids can be taught to pull that back and clean it carefully. About 15 years ago, the Journal of, I don't know if it was Reproductive Medicine or whatever, came out and said that there were statistics didn't show any advantage for males to have the prepuce, the circumcision. It's a little difficult to say. You'd have to talk to an adult who had had intercourse with and then had a circumcision and had it removed to see if there was different sensations. Because I bet my two boys who have been circumcised aren't going to tell you once they start having intercourse, which was better because they will never have experienced it with and without. But about 2 months after that article came out saying that for males it really didn't make any difference, there was an article or a letter in the Journal of Obstetrics and Gynecology that said that for women there was a decreased incidence of cervical cancer if they had intercourse with men who were circumcised. And that again is just back to the cleanliness issue. Okay? So if you have intercourse with a guy who maintains a clean area around the meatus there, that shouldn't be a factor. So a few months ago or a couple years ago, I don't know how long it's been, San Francisco tried to outlaw circumcision. That's San Francisco for you. But nowadays, it's a matter of, parental decisions. So, we made it for our boys before they had a chance to decide, and they haven't complained to me yet, but they probably wouldn't talk to me about it if they wanted to. So on Wednesday, we'll talk about female circumcision. It's not just the prepuce, which we have as females, but it's also their erectile tissue as well. Alrighty, so that's lab for today, and that 212 to 215 is what your quiz will be over on Wednesday as well.
So,